Awesome, cool, thanks. Yeah, so thanks so much for inviting me to the talk. It's really fun workshop. So I'm going to be talking about multi-agent foraging across different real-world environments. So multi-agent foraging happens across the evolutionary tree. This is a simulation of some slime mold in a single-celled phase following chemical trails together to get food. Probably what my ancestors, some of them imagined me doing with my life was foraging for food. This is an example of some people foraging for mushrooms. People forage in all different kinds of contexts, have for years, kind of all animals forage for food, often in groups. And there's a lot of changes in recent history, at least evolutionarily, about the environments in which we are doing these behaviors. And so this is just an example of a city, but a lot of these environments would be unrecognizable to our ancestors, like evolutionarily. And so I'm interested in characterizing these environments because it influences the behavior and the neuroscience really a lot. And just to get into like how some striking examples that got me into focusing on the environment is, so I did my postdoc in the Aronov lab, as Ching mentioned, and for those of you who have recorded place cells, this is a bird. Often see place cells in rodents, but the animal is moving around in an arena with a trajectory that looks like this. And you really want them to be moving to cover the full space of the arena so that you can see them going to each place repeatedly and get things like firing patterns in that corner for a particular place cell. And that is characteristic of this kind of featureless environment, but that's not the kind of trajectory you get in a lot of more complex and real world environments. When I was in my postdoc, here's an environment that is just slightly more complex that we designed. It has perches arranged in a circle. And so think about like what type of behavior would you expect animals to do in this environment? They can move anywhere. There's no constraints about where they can move in this environment, but think about like where would they move in the environment? And it's pretty striking. I should try to move this. Oh, great. Okay, cool. So each dot is where the animal was at a different time in the experiment, and the bird is really focused on these perches, which are most comfortable and ergonomic for it, and doesn't really go to other locations much at all in the arena. And this also shapes the hippocampal activity. It's still a place cell. You can still kind of maybe predict what it would do from the more from the open field cases, but it really is shaped by the structure of the environment. And I was interested in getting ways of characterizing the environment instead of imposing the environment on the animal. And so I'll get into how to do this, but this is a 3D environment of where a lot of birds actually forage on these feeders in the forest. And this is a 3D model of this environment. This isn't a drone video. And so the idea is to capture these environments and then use them in models of multi-agent models of the behavior. And this is me at this field site with my new daughter, who I'm very thankful that she is letting me do all of this. So, and so what I'm going to discuss today is a motivating example of New York City rats. So like humans, rats in New York City are in environments that are quite different from a lot of their evolutionary history. And so that's a really interesting case there. And also we know a lot of neuroscience about the rodent hippocampus. And so I'm going to get into how I'm starting to quantify the environment statistics. And then I'm going to talk about methods that I'm using with other folks at Bases about inferring the behavior strategies of these animals. So this rat project is in collaboration with Ralph Peterson and Ahmed El-Hadi. We were doing some fieldwork recordings in various sites in New York. This is in the Central Park Ramble using thermal cameras, ultrasonic microphones, and RGB cameras to record some of this data. So what type of environments do rodents forage in New York City? They forage on the street. It's mostly at night. That's why these are darker. They forage on the street, near trash cans. They forage in the parks, and they forage in the subways. So here is an example of a thermal camera recording of groups of rodents foraging in Central Park. And we're using thermal cameras because they are easier to see, especially at night. They're warm, the background is cold. And so with this type of data, it's susceptible to computer vision tracking techniques. So this is using the segment anything model, SAM2, from, I guess, Meta, and each rodent is colored by an extracted footprint of that rodent's location. And so what type of environments are we seeing these rodents forage in? Again, you don't see much just looking at RGB camera, but with the thermal camera, you can pick up in the parks, groups of rodents foraging on the streets. Here are rodents foraging near a car, trash cans, etc. They're doing some kind of interesting types of behaviors in the subways as well. For example, these guys, those guys. And you can even see with some occlusion. So here there's a fence here. Again, it's shadowed, it's slightly occluded by some foliage, so it's potentially hard to see, but you can resolve where the different rodents are. So here is just some tracking examples superimposed through a couple minutes of the video. You can see that they're kind of aggregating near this patch where there was some food. And here are trajectories that were tracked through the video over time of rodents entering the frame and leaving the frame. So how do we capture this in this type of environment computationally in a way that's susceptible to our agent-based models? I'm relying on this Gaussian splatting technique, where the world is captured as a collection of tiny Gaussian blobs, maybe thousands of them, and the best fit Gaussian is captured from camera data. So the pipeline for this, creating this type of model, involves capturing pictures from many different camera angles using the call map algorithm to infer the intrinsic and extrinsic parameters of the camera. So this is where the cameras are and where different features that are shared across the different viewpoints are located. And then by placing Gaussians in each of these viewpoints, you can reconstruct this type of 3D scene. And here's just an example of a 3D scene from a subway type of environment. And you can start to quantify some of the statistics of these scenes. So in this scene, taking a top-down view of it and looking at the standard deviation of the centroids of the Gaussians in the Gaussian splat. And so here you can see the trash can, the fence, some foliage, some more open field cases. And we know that rodents prefer shelter and don't like open field. And so you could look at this and say, well, the sheltered ones have higher standard deviation in the z-axis, and those are more appealing locations compared to the flatter, more open field type of environments. So now we have an environment that looks kind of like a grid world environment that we're used to modeling things in, but it's actually extracted from some environments that the animals are actually foraging in. And this is just that same type of analysis from one of these subway models where you see like the edges of the subway are more sheltered areas compared to the tracks, which are more open field and scarier for the rodents. And this idea of capturing the statistics of natural images is kind of an old or classic inspirational idea in neuroscience. So Olshausen and Field use natural images to extract using sparse coding these type of features from natural images, and these look very much like receptive field properties of single cells, but they're extracted from natural images. So that's just saying like a lot of what we see in the brain is actually due to the statistics of the world. So it's important that we study the statistics of the world. And then there's also really inspirational work from Kim Stackenfeld and others about the hippocampus as a predictive map showing that different types of tasks actually give rise to the type of firing patterns that you see in the hippocampal formation. So there's really a big dependency on the type of environment and tasks that animals are operating in. And then I wanted to mention, I'm also getting this data in addition to the rodent project, a variety of places where birds forage. So I went to Arctic Alaska. This is a couple 100 miles south of the Arctic Circle, just showing how I'm acquiring the data in a way that doesn't scare the birds too much. Otherwise, we would use drones. And it was at Toolik Field Station, which is a really, really wonderful field station in the Arctic Circle. And here's an example of one of the Gaussian splat models of the Arctic tundra. So this is an environment where large groups of migratory birds are flocking and foraging for food before going even further north. So finally, I wanted to get to some work that we've been working on inferring different behavioral strategies from this type of movement data. And this is more in simulation now. So I guess this is all quite preliminary, and I'm really excited to work with many of you in the future on developing this. But this is all after saying we should move beyond grid worlds, we should do all of this natural environments. This is using simulated data in grid worlds to try to nail down some of the more quantitative methods for inferring this stuff. So this is together with Rafal Urbaniak and Marjorie Shi, and we realized that there's a lot of different perspectives, each with their own kind of notation for studying multi-agent animal behavior. So there's more of a cognitive perspective with these interpretable RL models that captures in the value function what is driving animals' behavior. There's more of a statistical perspective of trying to predict what their behavior will look like. And then there's more of a neuroscience perspective of what might be happening in their brain to give rise to these behaviors, from like sensory processing all through motor action. And I'm not gonna get into the notation so much in this talk, but it is published, and we were able to show that there are similarities between the notations that are used in this field. So we can actually kind of map between the different notation. So in the cognitive perspective, if this is a map of an environment where this is a value function, so there's food here, and value diffuses there, you might say birds value food. And from a neural perspective, you might say the brain computes a food value trace. And then from a statistical perspective, you might say that this trace here is a good predictor of where the birds are going to move next. And so what we did was phrase different hypotheses as terms in a value function, but then use statistical inference to predict or to fit which ones best predict a particular type of behavior. And we can decompose a value function into different pieces. So this is a value function, which is the sum of wanting to be near food and also potentially wanting to be near other agents in the group as well. So we've simulated different types of agents. There are agents that move randomly. There are agents that we call them the hungry agents, where they didn't care about any other agents in the environment. They just approached food. and then the follower agents didn't care about food and then just cared about being close to other agents. And we wondered whether we could infer the strategy from the behavior. And so 5 minutes, okay, cool. And so we developed different predictors for each of these hypotheses, the hypothesis that they care about being close to other agents, or the hypothesis that they cared about food. So here, each location that this red agent might go is shaded by how likely that hypothesis says that they will go. And so under the proximity function, proximity hypothesis, this agent is most likely to go to these preferable locations around these other agents. And under the food hypothesis, this agent is most likely to go to locations that are near food with an exponential decay similar to what you'd find in the successor representation. And we fit coefficients to these different models and found that the posterior predicted distribution of the coefficients for the random birds was near 0, but for the hungry birds, there was a higher food coefficient, and for the follower birds, there was a higher following coefficient. So we're able to use this type of model to profile different types of strategies that animals might have. In general, at BASIS, we're trying to build off of these causal probabilistic programming tools where we express different strategies as different programs can take in heterogeneous types of data, different types of hypotheses, and then do Bayesian inference on them. So we did posterior inference of what the different parameter ranges should be. We want to be able to do counterfactual queries, like what if the environment changed in this way? How would have that been different? Different types of inference in that framework. And so we've collected, or we posted this code in a repository that we're calling Collab Creatures on GitHub. And this is also drawing on some of the other work from others at BASIS with Pyro and Cairo, which are causal probabilistic programming frameworks. So yeah, it would be great if anyone has a data set that they're interested in trying some of these tools, please reach out. I wanted to thank my collaborators and advisors on this project and funders and also my daughter and my husband for making this possible so short after she was born. So, yeah. Thanks. Go ahead.
I find some of the ideas you're reading. And question I always have about this outdoor naturalistic approaches is how do you feel about being able to manipulate it? Like I know that the modeling perhaps around how different I'm not having it. Not necessarily using experimental control, but in control of the environment by definition. Right?
Yeah, that's a really good question about manipulating the environment in these natural environments. I think that there's kind of two possible approaches I see. One is that there's often kind of natural experiments where you can manipulate the environment, at least like where food sources are. With the New York rats, you could imagine like they're already doing manipulations of like removing a dumpster of food or cleaning up this area. And so that is something to just like focus on cases where those are happening and look at the causal effects there. And then the other answer to that is to try to use these kind of causal probabilistic programming methods to model the different causal arrows between different cases and then use that to try to predict what might happen in cases that you didn't actually see in the data. Yeah.
Actually, Don, you can go ahead. I have the numbers, but you showed me like the.
Yeah, it kind of just depends on like what cameras you use and how zoom. It can be quite large if you can see it with the camera and that you can see a certain point with multiple camera views and you have good enough resolution. I don't think it's like that constrained on how large it could be. Yeah, so like if you, because you could look at like a miniature environment versus a huge environment, you're just like farther away with your cameras in the huge environment. So your resolution might not be as good, but it depends on your camera resolution a lot. So yeah. Mm-hmm. I think probably, and I think different animals probably have different capacity to do that, which I think is a really interesting question. Like some of the birds that I was studying in my postdoc have enlarged hippocampus, like birds in the chickadee family, and so they probably have larger capacity for forming a model of each other than birds with smaller hippocampus. So.